Well, good afternoon to everyone. Thanks for being here after a hearty lunch, I hope. When we look specifically at stress and cancer progression, like we can do with this forest plot from the meta-analysis by Andrew Steptoe's group, we see that almost all of the cancers examined are carcinomas, a total of 134 studies that they looked at. Compare that large number to the relatively paltry amount of studies looking at hematopoietic cancers, and when you do that, you can see that relatively little examination has been given to leukemia. Given both the scarcity and the role of leukemia in overall cancer mortality, we decided back when we started this line of research that we wanted to take a preclinical model of the disease to test for a causal relationship between stress and leukemia progression. To accomplish this, we used the Xenograph Model. We started with 10 SCID mice and injected them intravenously with human pre-B-cell acute lymphoblastic leukemia for subsequent bone marrow engraftment. We then randomized the mice to either a home cage control condition or to a restraint stress condition where they underwent 2 hours of daily restraint for 14 consecutive days. We then measured leukemia tumor growth and dissemination at days 7, 14, and 21 with a CCD camera system that is able to detect not only where the leukemia cells are in the mouse, but also to count how many there are, relatively speaking. The camera can do this because we transduce the cells beforehand with firefly luciferase gene. It's the same gene of fireflies that enables them to glow at night, for those who don't know. And these colors here that you can see on the graph, they represent the relative amount of light being given off by the leukemia cells, measured as photons per second. So what did we find when we did this? We ran this experiment 4 times, and here is a representative picture of a control mouse in this model showing 3 hot spots, one for each femur and one up in the thorax area. Compare that to a restraint stress mouse that has much heavier progression but still in similar areas. And when we look at the group means quantitatively, you can see that there's not much going on in this model at day 7, but that stress begins to show its effect by day 14, and then this effect continues at day 21. So in response to our first question, the results seem to indicate that yes, chronic stress can accelerate leukemia progression. But how is it doing this? Well, as we all know, being in this room, doing the kind of work that we do, when it comes to stress and cancer progression, neuroendocrine mediators that get looked at include catecholamines from the sympathetic nervous system, including noradrenaline/norepinephrine. And we know that sympathetic nerve fibers, do innervate the bone marrow where leukemia cells reside and could possibly affect the leukemia cells through that pathway. In fact, at the time when we began these studies, it was starting to be shown that beta-adrenergic signaling does presumably play a large role in the sympathetic nervous system in other preclinical models of cancer. So with this in mind, we ran a new set of experiments to answer a second question. Does chronic stress enhance leukemia progression through beta-adrenergic signaling? For this study, we took a group of 20 SCID mice and inoculated them with human leukemia as before. They were then randomized into 4 groups of a 2-by-2 experiment, home cage control plus saline placebo, home cage control plus the beta blocker propranolol, and then the stress condition plus placebo and stress plus propranolol. We then again monitored tumor load via bioluminescence as before. So what did we find with this group of studies? Well, first you can see here that there was no significant difference between control mice and mice receiving placebo or control mice receiving propranolol. However, here in the second half of the figure, you can see that propranolol is effective at blocking the stress-enhanced leukemia progression, similar to what we've seen in some of the other models of carcinoma progression that have come about. So coming back to our second question, the results seem to indicate that yes, the effect of chronic stress on leukemia progression does seem to be mediated through beta-adrenergic signaling. But how so? Does norepinephrine stimulate the leukemia cells directly? That answer seems to be no, because as you can see here in this graph, we ran proliferation assays where we stimulated the leukemia cells directly in vitro with norepinephrine and then checked for differences in the proliferation at 2 hours, 6 hours, 12 hours, 24 hours, all the way out to 4 days. And we didn't see any effect. Also, when we tested for the functionality of beta-adrenergic receptors on these cells, we found no results as evidenced by a lack of any second messenger cyclic AMP flux within the cell, although the cells were certainly capable of producing cyclic AMP as indicated in our positive control with forskolin. So we began to consider possible indirect pathways by which beta-adrenergic signaling could exert its effect, which included CXCL12 regulation. I don't have that data with me, but we did investigate and found that CXCL12 was upregulated in the femurs of these mice when we looked at it by ELISA. And we know that Paul Frenette's group, which is now at Albert Einstein College of Medicine in New York, he had shown that sympathetic signaling can regulate CXCL12 chemokine expression in the bone marrow as well, and thus alter the behavior of CXCR4 positive hematopoietic stem and progenitor cells, which are very similar to the kinds of leukemia cells that engraft inside the bone marrow. Thus, one potential indirect pathway may involve, we thought, adjacent stromal cells in the bone marrow that support leukemia cell proliferation, such as osteoblasts that are being depicted here in this diagram from Paul Frenette's group. So with that in mind, we ran a new set of experiments to answer a fourth question. Does CXCL12, CXCR4 signaling mediate the beta-adrenergic effect on leukemia progression? Once again, mice were randomized into 4 groups of a 2x2 experiment, but this time, instead of using a beta blocker, we used the CXCR4 receptor antagonist, AMD 3100, and then we measured for bioluminescence as before. So what did we find? Well, we struck out. Here you can see that the group receiving both restraint stress and AMD 3100 had just as much tumor load as the group receiving restraint plus placebo, maybe even a little bit more. So coming back to our fourth question, the results seemed to say at that time, no, the CXCL12, CXCR4 axis is not a downstream mechanism of stress-induced beta-adrenergic signaling. So we then turned our attention to macrophage biology, given the connection that has been found between increased sympathetic signaling and increased tumor-associated macrophages, as can be seen here in Erica Sloan's breast cancer work at UCLA. We thought maybe sympathetic signaling is causing more macrophages to accumulate in the bone marrow to help the leukemia progress, like it seems to do in breast cancer and other carcinoma models. So with that in mind, we ran a new set of experiments to answer a fifth question. Aside from stress, does macrophage inhibition by itself reduce leukemia burden? No one had really looked at this yet at that time. To answer this question, mice were randomized to either a placebo control condition or to a condition where they received a drug called GW2580, which blocks receptors for macrophage colony stimulating factor, MCSF. It's also known as CSF1 or colony stimulating factor 1. So then what did we find here? Well, here you can see that contrary to our hypothesis, the group receiving GW2580 actually had more tumor load than the control group, which kind of befuddled us a little bit. Coming back to our fifth question, then the results seem to say that no, macrophage inhibition does not reduce leukemia burden. In fact, macrophage inhibition seems to increase leukemia burden, at least in this model. So what's going on here? Well, it may be helpful to understand that macrophage function can be quite dynamic depending on where it falls in the so-called M1, M2 polarity spectrum. Macrophages on the M1 side are seen as antimicrobial, pro-inflammatory immune cells with growing evidence, especially in the immunotherapy literature, that they increasingly are facilitating anti-tumor immunity. In contrast, M2 side macrophages are associated with immunosuppression, tissue repair, and most likely facilitation of carcinoma invasion and metastasis. And recently we found, using a genome-wide approach, that beta-adrenergic signaling causes macrophages to not only upregulate M2 gene transcripts, but it actually causes them to be actively suppressed of their M1 gene transcripts. So, thus, now we're preparing to answer a new question. Does stress-induced beta-adrenergic signaling suppress M1 gene transcripts in bone marrow macrophages, making them less capable of being anti-leukemic? And we have additional pieces of evidence to suggest that it does. Among the 113 genes in macrophages that were suppressed by beta-adrenergic signaling was a gene called Siglec1. It's the gene for CD169, which is becoming more associated with anti-tumor immunity in the research literature. And sure enough, when we look at mice receiving daily isoproterenol injections, we find that macrophages from bone marrow are expressing lower amounts of CD169 phenotypically. Thus, our new hypothesis is that suppression of M1 properties in bone marrow macrophages may be a downstream mechanism by which stress-induced beta-adrenergic signaling increases leukemia, a hypothesis that we are planning to examine in the near future. So to conclude, I want to acknowledge the many persons and funding sources that have supported this line of research, with special thanks, of course, to the NCI Network on Biobehavioral Pathways in Cancer. Thank you.